So we are going to run a viability experiment, and I've set up all the things we're going to need. So we need our cell suspension. I don't like going into any of my stock bottles, so I've created a small tiny 1.5 ml tube of all of the things that we're going to need, little aliquots. And so if you follow along with our manual, our protocol, we're going to start with the cell suspension. So now I have to be cognizant of the fact that the cell suspension is something that I want to keep sterile because I'm going to be plating my cells from here, but this experiment isn't going to be done sterilely. So I'm going to take a little aliquot of cells out. And once I've done that, I will use those cells to create a dilution, a 0.5 times 10 to the 6th dilution. And I did all my calculations and I calculated that I needed 16 microliters of cells. And to those 16 microliters of cells, I'm going to add 84 microliters of PBS. And so now I'm going to set up my experiment. I have a cell suspension that has 500,000 cells per mil. I have 3 tubes, one labeled control, one labeled 1, and one labeled 2. And to each one of these tubes, I'm going to add 20 microliters of cells. So whenever I have my pipette in my cells, I do pipette up and down a few times before I grab my sample, just to make sure the cells have not settled. I also then just kind of drizzle the cells down the tube rather than settling them on the bottom. I don't like to create a small pocket of air at the bottom of the tube, which could cause me problems later. So now I've added 20 microliters of my cells to each one of these tubes. You'll notice that that liquid drizzles down happily to the bottom of the tube. I will now add 20 microliters of each one of my treatments to the appropriate tube. So I'm going to take 20 microliters of PBS and put it into my control tube. At this time, I'll shut my PBS and I'll move it aside because I've just added it, and I'll close the tube that I just added something to. I will then change my tips and I'll add 20 microliters of my water to my tube experiment number 1. So I'll close my water up and I'll move it aside. I'm done with that, and I'll close my tube number 1. I'll then change my tip and I will grab 20 microliters of my hydrogen peroxide and put it in to my tube 2 experiment. And now I will have my experiment going. Okay, I am going to finger flick, that's hard to do with one hand, each one of these samples and then let them sit for 10 minutes with their appropriate treatment. And when the 10 minutes is done, I'm going to put 20 microliters of Trypan blue. So Trypan blue is a solution that gets into the cell, and if the cell is dead, it can't get it back out. But if the cell is alive, it can take the Trypan blue and move it out. I don't like going into my stock solutions, so I've created a tube of TB. So my experiment has gone for 10 minutes, and so I'm going to drizzle 20 microliters of Trypan blue into each one of these experimental samples. C is a control, and all of these cells were in an isotonic phosphate buffered saline, so they should be okay. They shouldn't have died. Number 1 had water for 10 minutes, and number 2 had hydrogen peroxide for 10 minutes. Once again, Trypan blue will make the cell blue if it is no longer viable or it has died. And so what I'm going to do is pipette up and down and get a 10 microliter drop of each one of these samples